EPO, otherwise known as erythropoietin, is a protein that increases the production of oxygen-carrying red blood cells. EPO is released from the kidneys and acts on the bone marrow to increase the production of red blood cells, which increases the amount of oxygen that can be carried to the muscles and other parts of the body. EPO is most well known as one of the enhancement drugs of choice for Lance Armstrong and other endurance athletes. Research into the identification and uses of EPO began in the early 1900s. Building on initial ideas, researchers established that low blood oxygen increased the release of red blood cells through the EPO hormone, and that the kidneys produce EPO naturally. The EPO protein was first used as medicine to enhance the blood of patients that underwent chemotherapy for cancer. Another early use was for treating the effects of kidney disease by repairing damaged blood as a result of dialysis. In the figure shown, EPO therapy for anemic patients vastly increased their measured red blood cell count. Manipulating the levels of EPO was later discovered to enhance the performance of athletes. EPO enhanced performance by increasing the amount of oxygen going to the muscles of athletes, thus increasing their endurance and overall ability. Essentially, EPO provides athletes' muscles with more fuel. According to Dr. Schwartz Bloom at Duke University, endurance athletes who use EPO can increase the oxygen-carrying capacity of their red blood cells by as much as 7 to 10%. However, if EPO is abused, it can cause blood thickening that can lead to heart disease, stroke, embolism, and even autoimmune disease. Although EPO is produced naturally in the body, it can also be produced artificially with bioengineering techniques. Recombinant EPO is produced with the molecular cloning technique called genetic recombination to produce a recombinant protein. As Dr. Jang of Virginia Tech explained, it is important to realize that EPO is a protein and cannot simply be synthesized in a beaker like synthetic chemicals can. Proteins are made by recombinant methods using a bacteria such as E. coli. The code of a protein must be expressed, then purified, and purification is a significant engineering challenge. Dr. Sanger of Virginia Tech elaborated that in engineering recombinant proteins, human cells are not used in the artificial engineering of proteins. Looking ahead, studies show that EPO can be an important molecule for the protection of cells and as an anti-inflammatory agent. Researchers are investigating EPO as a potential treatment for a multitude of diseases, such as Alzheimer's disease, Parkinson's disease, and psychiatric disease. It also has the potential to be used in the aid of DNA repair and even stem cell development. The study of EPO is a complex engineering challenge that future biological systems engineers can advance.